Cellular homeostasis, proteins. The objectives are to identify the different levels of proteins, explain how protein structure determines its function, explain the process of protein sorting, describe protein synthesis and breakdown, explain how protein functions are regulated, and to describe how homeostasis affects protein folding. Protein structure and function. Proteins are composed of carbon, hydrogen, oxygen, nitrogen, and small amounts of sulfur. Proteins are macromolecules that play critical roles in most life processes. The proteins of all living organisms are composed of the same set of 20 amino acids with different side chains, as you can see in this figure. Amino acids are joined by linking the carboxyl group with the amino group of another amino acid, forming a peptide bond. And as you can see, this is also a dehydration reaction. A polypeptide is a structural unit composed of amino acids, whereas a protein is a functional unit composed of one or more polypeptides that have been folded into a three-dimensional shape. 4 levels of protein structure. The 4 levels of protein structure are primary, which are just the amino acid sequences, secondary structure, which includes alpha helix and beta pleated sheets, tertiary structure, which is just a three-dimensional shape, and quaternary structure is when one or more polypeptides come together to form a structure. The three-dimensional structure of a protein determines its function, for example, creating binding sites for other molecules. Okay, protein sorting and specific targets. Proteins contain short stretches of amino acid sequences that direct them to their correct cellular location. These sequences are called sorting signals. Each sorting signal is recognized by specific cellular components that direct the protein to its correct locations. Proteins begin their synthesis on ribosomes in a cytosol using mRNA that contains the information for polypeptide synthesis. The cytosol provides amino acids which are used as building blocks to make these proteins during translation. Cytotic proteins lack sorting signals, and that's because they stay in a cytosol. By comparison, the synthesis of some proteins begins in the cytosol and then stops for a minute until the ribosomes bind to the ER membrane. After this happens, translation continues and the polypeptide is synthesized into the ER lumen. And proteins are destined for the ER, the Golgi, the lysosome, vacuole, plasma membrane, or secretion. And here we see that the proteins are sorted in a Golgi apparatus, and then they are budded and by Blebin, and then they interact with the receptors of the lysosome, and then fuse with the membrane of the lysosome. And then we can see also here we have the cytosol, the proteins in the cytosol, and we also see that some proteins are secreted. Protein metabolism. Well, proteins can be broken down into amino acids, and once they are broken down into the amino acids, they can be absorbed into the bloodstream. And then when you break the amino acids down, the nitrogen or the amino group is removed from the amino acids. And then they can be broken down into ammonia, which can then be converted to urea, and then they can be filtered through the kidneys or excreted in urine. And sometimes amino acids are broken down and they are called glucogenic amino acids, which can be made into glucose. Protein function and regulation. Well, most proteins function as enzymes, And protein activity can also be controlled by the binding of effector molecules, which often work by inducing conformational changes that produce active forms of the proteins. And effectors may bind covalently or non covalently, and this modifies the structure of the protein reversibly or irreversibly. Effectors that regulate activity by binding to the active site usually take the form of inhibitors that compete with the substrate for binding, and these can be competitive, non competitive, or uncompetitive. Proteins and homeostasis. Well, we know that structure determines protein function, and homeostasis is required for proper function of a protein and proper folding of a protein. Proteins rely on the correct folding sequence to form three-dimensional structures capable of performing their intended functions. Disruptions to this homeostasis can lead to proteins that are abnormally folded, which can change the way that the protein functions, and even in some cases cause diseases to happen. So, genes have a major influence over the maintenance of protein homeostasis. Expresses expressing genes or allowing them to interact with other components of a cell to form proteins is one way this can be accomplished. Proteins Protein homeostasis is maintained by cell signaling. Well, cells must be able to respond to a variety of new conditions, and protein homeostasis plays an important role in this process. Signals from the environment can induce the creation of proteins that allow for cells to manage these situations. And these signals can be issued from inside of a cell, from an organ, or from a group of organs, depending on the extent of the changes that the organism is responding to. Also, protein homeostasis can be maintained through feedback mechanisms. Feedback mechanisms also helps maintain protein homeostasis, and it also involves multiple organs. Enzymes and cellular structures interact with newly made proteins to ensure that they are folded into their correct conformations. Information on this folding must be sent to the nucleus of the cell or even to the brain, which can then send back return messages about further actions to take. Sometimes irregular proteins may lead to feedback signals that orders them to be destroyed. And this concludes the protein section of this presentation. Thank you.